So, good evening, bachcho. Very good evening. So, we were lucky. I just wrote you message, and suddenly electricity came in just after 1 second. So, the proof that I was sending on the Telegram, the reason behind it was our administrator is also looking what we are doing. So, in order to do so, we need to have proof. Why things are not working. Because if I started 14% and my laptop would discharge within 5 minutes, and then it doesn't make sense to start the class. But we are lucky enough. Now it's 21%. That's good. Um, so I'm having a lot of people are calling me. A lot of havoc is going on. Oh, I saw this picture. Maybe I should share with you from I Love PhD. So when you're writing a review paper, the references amount in that oil should be like this, not little but a lot. So and have this happy face while doing so. Yeah, so this should be the target of us. So something funny, um, I could share with you so that we can remember this funny joke. Uh, So there are some students, they're asking me doubts, so I will reply to them here. So I cannot just reply one-on-one on the messages. So I'm gonna reply here. If you're online, please go through it. So Nishta Madan, we are in BSc, we haven't written any review paper till date. And it's like we're not able to understand how to write it. Can you please help us? Nishta Madan, when did you join this? I think you joined on 2nd of February. From the first day you are there, you're asking me on the last day this question. It's very sad to hear about this. Please, it's too late now. Should have asked before and joined one of the review group. So this is not the way you will ask on the last day of your day of your work that when we have what is it all about. It's not the way, please. So I have written my my paper on golden rice. Is that okay? It will be okay if I can see it once. So hello, is somebody even called me? Devasmita Banerjee. Devasmita. I am doing MBBT. I have questions. When we give the review paper in between 26th February. So I mean, you can send me before 28th of February is the date. If you can, the more early you send, the more early you get the certificate. Yeah, before 28th February. So what's the next question? Hello sir, MBBT. Hello sir, I have done my review till now. I have like to help like your. Why are you asking on the last day? Could you please let me know the topic I should select and what it will be written? Someone pinned the old message in the group. Thank you, sir. Okay, you got the answer, Rutuja. Sir, this is Sneha Roy Chaudhary, a participant of MBBT. Abhay Shrivastava and myself, we work together, computer paper. I have mailed the same to. Okay, so you have mailed me. Okay, that's good. Sanika, hello sir, I'm first year. I have opted for internship option. Can you please explain? Bache Sanika, how can I help you on the last day? Help you on the. Where were you before? This is not the way. Please. Somebody could help them. Okay, I think that's Ipshita Basak. I'm Ishita Basak was submitted a paper. If three groups are doing a single review paper in a group, when will the members have to mail the review to separately? Yeah, separately. Last date 28th. Feb. So anyone has question here? And review paper group will have to submit separately. One person. I just answer your question there. Okay, sir. But you have to Nishta Madan. This is Nishta. This is your first message. You can see it. Okay, sir. Thank you, sir. Today on 23rd of Feb from the 2nd Feb, I got your message today here. So please ask some group if you can be part of it. Only then you can go ahead with it. Otherwise, there's recorded lectures. You can watch them. You can watch them and look for it. Okay, good. So we start the lectures. So let's start. Yeah, Anjali, we might have some MCQ type of questions. I'm trying to convince our editors there. So let's see. So something about writing review. You will be doing research in future. How to practice good scientific practices in science. How to do work in science. So here you can see prominent cases of that. So there are a lot of people who do some bad things in science. They refabricate your results. They do not make things done as it should be in order to do so. So there are many things are happening in the scientific world, like retraction of papers are happening. Regression of human metastatic renal cell carcinoma after vaccinations with tumor cell dendritic cell hybrids. This is all fake, no? This is all fake. So a good scientific practice should have ethical approval. You should have maintain your original data. So these are the various FAQs, like frequently asked questions. Can you copy a sentence from another paper? Under which conditions can I use from another paper? Who qualify for the co-authorship on my paper? And whom should I contact if I suspect scientific misconduct? So these are the various questions that comes into the mind when you want to start your research. What matter what and how things should be done in a way in order to do the things. So responsible conduct of research. So values, legal framework, institutional policies, data management, and lab notebook keeping. So these things should be maintained properly. Then a case study has to be there, and then a misconduct in research like fabrications, falsifications, plagiarism has to be there also. Then shared values for the responsible conduct of research. Honesty has to be there with proper accuracy, efficiency, and openness. So whatever the information that we are providing, are we conveying? Is it truthful, honoring, and committed? Then in accuracy, reporting finding precisely and taking care to avoid errors. And then efficiency, using resources wisely also. Then openness, letting the facts speak for themselves, avoiding improper bias in that. Then good scientific practice has to be maintained. What are the various points under the general principle of it? The first section, the rules that should be respected, the things should be preventive measures. There should be no scientific misconduct, and contact the person if somebody did this scientific misconduct. And in the procedure of suspected scientific misconduct, contact your university or medical center, investigate your committee that under you're working, and then should be formal investigation of the investigating committee. So common reporting, additional measures, storage of records has to be taken care of. Then protection of human subjects, local ethics committee. So participation must be voluntary, special consideration for vulnerable subjects, and there should be proper beneficence, obligations to maximize benefits, obligations to distribute benefits and risk equally without prejudice to particular individuals or groups. Then welfare of laboratory animals, animal protection committee, replacement, reduction, refinement. So using non-animal models such as microorganisms or cell culture techniques. Then reduction, using methods aim at reducing the numbers of animals such as minimizations of variability, appropriate selections of animal models, and minimizations of animal loss. Then refinement, the elimination or reduction of unnecessary pains and distress. What are the rules of good practice for doctoral supervisions? So doctoral students should be informed about the supervisor immediately if there's any problem or unexpected results are happening. And supervisor should be confident of the primary contact person for the doctoral student. And thesis committee that you're having should be, there's no instance of conflict between your doctoral students and supervisor. So these are the things that you will be doing research in future. You might be mostly from bachelor's or master. Some of you might be a PhD. But these things will matter for the things. But for how do you get our own pictures for the review? In my case, in my PhD thesis, this was my PhD defense. So I will tell you one example. So these are the images. I made it by myself on the PowerPoint presentation. So I choose this CMP, pKa, all these various figures by myself, and this heart pumping figure, and making these things on my own, and like this also. So whatever the figure here, I have adapted it from the Meng et al. Journal of Cancer Molecules, showing the glutathionylation and everything in it. So some of the figures, these are the results that I have discovered. So in most of the cases, this is one I created. So whatever the figure that I see in book or anywhere, I used to make it on my own actually. That should be the priority. Never just take book from that. If you're taking, just write down that it is being adapted. Otherwise, it doesn't make sense. Yeah. On the other hand, so ER stress signaling pathway. So this is you can see that PERG, RE1. This is also I made by myself, like all these figures with the signaling pathway. So this was, I saw one picture, but I added many things by myself. So I just took like, I read 3 or 4 reviews and I added these pictures in order to understand about CTGF6. So this is how you have to do your things then. Make things on your own. That will be better. So further, doctoral students, supervisors, thesis committee members, university boards. Then data management has to be taken care with the data ownership, data collection authorized by using appropriate methods, hard copy evidence, and electronic recordings. Then storage of lab notebooks in safe place, backup strategy for electronic files. Then further data retention, primary data that serve as the basis of publications must be stored for 10 years. Then data sharing, primary data published results should be made freely available afterwards. And some rules for making your notebook. So this is what also very important when you are going to work in your lab. Your lab book has to be maintained nicely. With the notebook should be bound, paper durable, pages should be numbered, and must remain on site. Should never be taken from home for any purpose. All notes must be easily legible and permanent blue or black ink. Pages must not be erased from the lab notebook. So this has to be taken care. Then rules for good lab book notebook keeping. So date of entry, descriptive title, purpose, methods, results, conclusions of each practical that you're doing. Because at the end of the day, when you start your PhD or master thesis in a lab, there will be tons of data with you. They're very huge, like we were doing multiple RT-PCR, doing a lot of Western blots, doing DNA microarray, SDS page, some protein quantifications, or you're doing cell imaging, immunofluorescence imaging. You're doing many different kinetic assays, phosphatases assays, kinetic assays, so there will be many things, so much of data with you going on, but it has to be kept in order, it kept in a way that it should not be gone wrong at the end. So this will really help you to be a good scientist at the end. So this is the rules for the good notebook keeping. So here you can see providing with page numbers, session 1, session 2, title, plan, date, name of the lab partner, everything. Then scientists behave badly. So what is happening is there's a lot of pressure on the scientists at the end of the day. So they have to publish things and perish, publish and perish. If you can't publish, then you can't live in the science. You will be go through down. So to do so, there are some top 10 behaviors that we find in the carriers of people. So like falsifying cooking research data, ignoring the major aspect of a subject, not properly disclosing involvement in firms, relationship with students, research products, using another ideas without obtaining permissions, unauthorized use of confidential informations, failing to present data that contradict one's own previous research, overlooking others' use of flawed data or questionable interpretations of data, and then changing the design, methodology, or result study in response to the pressure from the funding source. And other behaviors, publishing the same data or results in 2 or more publications, inappropriately assigning authorship credits, withholding details of methodology results in the papers or proposal. So what are the misconducts in research as done? So they are such as fabrication is done, making up of data, falsifications, manipulating the changing or omitting data, plagiarism, like stealing another person's ideas, sabotage, adversely affecting the research activities of others, or violating the recognized rules of authorship. So these are the major 5 things that people are doing: fabrications, falsifications, plagiarism, sabotage, violating the recognized rules of authorship. So it doesn't think that the science is doesn't think that science will be like a bed of roses. It might be with a lot of thorns in it. Um, so believe me, if you're gonna start your career in research, be ready for a long marathon and working hard for it. Yeah, here we can help you. No worries, no worries. But remember, this is your own development. As you go studies, as you study further in your life, uh, you will be getting better of yourself. You will be a better version of yourself each day. Either you could be better version or the worst version. You have to choose yourself. Remember that part. You can choose either of them. There are 31 students here who are the lucky one who want to listen to this part. So believe me, these things will help you on the long run. So better make a strong will, make a strong mind in order to work for the science field in the future. So yeah. So fabrication is making up of inventing data, results, recordings, or reporting them. So falsification is manipulating the changing data, omitting the data. Here you can see there are some burglars in this minister Triton. So it has been shown that the Triton is carrying these people behind him, but he's not part of it. So there's a temptations of image manipulations, like you can change the gamma radiations of your plots, change the brightness, contrast, or color balance in order to understand the results. So here you can see that Adobe Photoshop Nonlinear adjustments of gamma settings being there present. You can see on the second one. Then here you can see some adding or deleting bands. So this is the original image. So somebody deleted the third band. So in order to get the results, this is JCP 2004 by Rosner. So this Rosner life is messed up for many years. His scientific career is being messed up now. And he added this band 3, manipulated like this. So first there was a band on top, then he deleted that band, and then he brought this new band. Then this is experimental 1A and experimental B. This is loading control A and B. So here you can see the image looks like this. In the B it looks like this. So it's all fabrication. Here the original image look like this, but the manipulation looks quite light here. So we are adjusting the bands here also. Also sequential in this fourth band, only this band is being seen, not the other things are coming up. This is original, this is manipulated. So yeah, Photoshop is doing everything. Some original image where you can see few spots of here, these spots, but in the manipulated you can see more spots. So again manipulated image will reveal that there is some contrast has been played out to show the more circumference of your cells. So these things please do not copy for your research, believe me. It's not good for you. This question, Bhupinder. So there will be one certificate you will get and one internship letter. So you will get two different things at the end of your work. One certificate of being part of that training, and one internship letter for being writing the review article. So this is plagiarism, is to steal another person ideas results without giving appropriate credit. So in this practice of taking someone else's work or idea, passing them of own, so be careful with that. The free use of copyrighted work permitted to bring about new independent work also. Plagiarism of correct quotation. So DNA in a eukaryotic cell is sequestered in nucleus, which occupies about 10% of total cell volume. So this is the plagiarism, that we copied the same sentence. And if we are putting like this, Albers et al. 1994 only, this is wrong quotation. And also paraphrasing is also, it's actually plagiarism. And this is direct quotation, like you're bringing everything here and then writing this. So this is also, but this is the right way to write it. In eukaryotic cells, the cells contains DNA and makeups approx 10% of cell volume. So this is okay. Yeah, this is also another version. So direct quotations, assertions, judgments, statistics, everyone has to be taken care. Some common knowledge, facts available in the wide variety of sources. These all things has to be also taken care. So yeah, that's it about the good scientific practice. Yeah. To this? No, not to Sherry Garewal. This is our last slide of today's workshop, of whole workshop today. So something what we have studied so far. Now we're gonna study about the applications of recombinant DNA technology in health, agriculture, environment, and industry. So these parts will be taken care now. Applications of recombinant DNA technology in health. So there's applications in health. So this recombinant DNA technology. It has a wide spectrum in improving the health. It treats defective genes and introduce new ones into them. And applications, laboratory test, parental diagnostic, genetic diseases, everything are being treated in this part. So first thing is production of vaccine. So we introduce live attenuated acquired immunity, and then this is protective antigen like hepatitis B vaccine, recombinant DNA, influenza, HIV, and mouth and foot diseases. So all these things could be taken care with the help of vaccines. So how do we produce it? So here you can see hepatitis B virus. We extract the DNA or RNA containing the HB antigen producing gene in that. And then we have a bacterium and we take the plasmid out of it, cut it with restriction enzymes, and we have a recombinant DNA at the end. Then you have introduction of recombinant DNA into yeast cells, and then you have recombinant yeast cells at the end with HB antigens. Then you have hepatitis B vaccines produced with the help of extractions and purifications of HB vaccine in that. So commercial and pharmaceutical products. So insulin controls glucose levels in humans, and by recombinant DNA, DNA cloning of human insulin gene and put in E. coli. Then availability of insulin and devoid of getting by products by the animal slaughtering. So in initial we understand about the producing hepatitis B vaccine. The same way we can produce the human insulin production. The same manner. Then human growth hormones, HGH hormone, being polypeptide, having 121 amino acids, role in the growth and regenerations or differentiations. An example is dwarfism treating by the injecting these. So here you can see that sequencing of 24 amino acids of HGH. Um, so you can see cDNA encoding for 24 amino acids, ligations for HGH along with the expressions with the vector genome and inserted into a vector like this. So interferon are a group of proteins that interfere with the viral multiplications or replications, and by recombinant DNA capable of making interferon. And alpha components which have role in curing lymphoma and malignous leukemia. Then there is antibiotic, artificial prepared antibiotics also available, and they denature harmful living pathogens. Penicillin in 1928, and then monoclonal antibodies being produced in our immune system in response to the specific antigens. So they are produced from the single clone of antigens. That's why monospecific in nature. And production through the hybridoma technology. So monoclonal antibodies are used for diagnosis of diseases, pregnancy, and treatment of cancer. So here you can see antigen is being immunized to the mice. We isolate these immune cells, this antibody forming cells. Then you have we put them into the tumor cells, we fuse them together, and then you have hybridomas. So these hybridomas are screened for the production of desired antibody, which produce hybridomas clone, and then the clonal expansion is done with the help of monoclonal antibodies. Yeah, I don't know. Akash Gurmi. You can choose any format like the ask Vatsal and Shivani about next. So other teachers are saying that authority told to take classes. I don't know about that. Sorry, Biplab. Do we need to give exam on this course? So I'm trying to convince that there should be one exam, but it not be compulsory, but it will be good for all of you to give it. So let's see how it will work. But how we conduct it actually? Do we have to be online or how? I don't know. Do you have exams online also? I'm not sure. How we will take your exam? Let's continue. Molecular diagnosis of diseases, infectious disease diagnosis maintains mainly depend upon the isolations and identifications of pathogens, which may take several days, and development of diagnostic kits to identify pathogenic organisms by knowing the organism specific DNA sequence, which has provided the rapid, specific, and correct diagnosis. So various diagnostic kits have been developed for AIDS, cancer, foot, mouth diseases, and tuberculosis. So based on ELISA and PCR principles, for example, in HIV patients, diagnosis conducted by antibody test uses recombinant HIV proteins to measure antibodies in the body that proliferate when there is HIV infection. So DNA test uses reverse transcriptions polymerase chain reactions to detect presence of HIV genetic material. And this technique was developed using recombinant DNA of molecules and analyze the genome sequences. So you have antibodies here, pathogen not recognized, pathogen recognized. So you have no binding and binding occurs on the other way. So based on that, you can do your ELISA and PCR. Then DNA fingerprinting we have discussed in detail to identify any foreign forensic deal used in the criminal identifications, child printer establishment for racial group. So you take your DNA, then restrict with the restriction enzyme. Um, then later on, you put your DNA on the gel electrophoresis, then bind to the nitrocellulose membrane. Then you see which DNA is working with that. Then there's a gene therapy which injects functional genes into a cell to replace missing or defective genes in order to correct genetic disorders. So gene therapy could be done in vivo or evo. So there should be some health risk of that, like toxicity, inflammation, and cancer. For example, you have a direct delivery of therapeutic transgene. The therapeutic transgene is packaged into a delivery vehicle such as virus and injected into the patient. So target organ, example liver, they have been ordered and re-administered to the patient. And the therapeutic transgene is packaged into delivery vehicles such as a virus here. And cell-based, on the other way is the cell-based delivery. You take your transgene into the cell and therapeutic added into your cells, then being cultured and then again re-administered into your organ. So that was about health point of view. Now in the case of environment, that is your bioremediations, environmental cleaning of water, pollution, waste, you can do it with the help of recombinant DNA technology. So we can use recombinant DNA technology in environment to clean up the environment from the hazardous compounds. So in this, for some harmful chemicals that are not digested or degraded, are partially degradable with the help of TNT chemicals, and is reacted with oxygen and superoxide is formed, that is toxic. Further, knockout of the monohydroascorbate reductase gene that will create resistance against this TNT. So arsenic particles, PVAC-R3 genes, PVAC-R3 genes, Arabidopsis thaliana, reductase enzyme, phytochelatins, they all resist the movement of arsenic in root cells and phloem cells. So waste product of agricultures have cellulose that do not easily decompose by using recombinant DNA technology and cellulose enzyme gene giving us vector. So recombinant DNA technology could also help in the plastic degradations, can be enhanced by genetically modified organisms. Then they could be degrade, oil spills, organic waste, and genetically modified strains of Pseudomonas proteida able to degrade chemicals in oil spills. They could be used in development of bioindicators, and bacteria have been genetically modified as bioluminescents that give off light in response to several chemical pollutants. Further, for example, mercury resistant gene or toluene degradation gene, TOL-MER, linked to the genes that code for bioluminescence and emitting visible light and measured with fiber optic fluorometers. So what are the applications of recombinant DNA technology in agriculture? So recombinant DNA technology can be used for insertions of genes in plants, not only from related plant species, but also from the unrelated species such as microorganisms. The importance, they are used for the productions of transgenic plants with high yield, nutritional values, increased resistance to stress and pest. Then there is a development of plants having improved yield, development of stress tolerant plant, and transgenic plants as a source of biopharmaceuticals. So genes are inserted into plants to increase their yield. So researchers at the Japan National Institute of Agribiological Resources added maize photosynthesis genes to rice. Increased yield of 30% has been seen with that. Then Mexican scientists have genetically modified plants to secrete citric acids from their roots. So in response to the slight increase in acidity, minerals bound to the soil particles. Then stress tolerant plants, plant resistance to environmental stress. So recombinant DNA technology is used to develop crops that can tolerate abiotic stress, and genetically modified tomato and canola plants that tolerate salt levels of 300% greater than normal. Then herbicide resistance plants. So there's a roundup of herbicide, but it kills almost all species of plants. So using recombinant DNA technology, modified EPSP synthase genes have been introduced into the crop plants such as cotton and soybean. Then insect resistance plants, cry genes from bacterium Bacillus thuringiensis are isolated, and then plant is modified using this gene. Example, cotton, rice, maize, potato, brinjal, cauliflower with the BD genes have been produced. Some disease resistance plants to create a resistance against diseases. So tobacco was the first modified tobacco mosaic virus resistance against plant have been produced, TCME49 here you can see, and this is a wild type. Then production of biopharmaceuticals to produce a plant that will generate a seed that expresses a desired therapeutic protein. Example, your corn, in corn we are producing biopharmaceuticals are produced in that case. So edible vaccines, so the genes encoding antigenic proteins can be isolated from the pathogens and expressed in the plants producing antigens can be eaten for vaccinations or immunizations. So in banana, tomato, edible vaccines are made. Productions of secondary metabolites. Arabidopsis plant is modified. Polyhydroxybutyrate is released by Arabidopsis, and PHB is being used as a biodegradable plastic. So some applications of recombinant DNA technology in industry. So traditional industrial microbiology is merged with molecular biology to yield improved recombinant processes for the industrial production. So primary and secondary metabolites, protein biopharmaceuticals, industrial enzymes are formed for the industrial usage. So important primary metabolites like amino acids, nucleotides, vitamins, solvents and organic acids, and their production is related to the energy production and substrate utilization, which are essential for the growth. Then vitamin B12 is being produced commercially by direct fermentations utilizing the fungus of Asbia gossypii. And vitamin B12 is produced by the direct fermentation utilizing Streptomyces species such as Streptomyces griseus. Further, it is being utilized in the gluconobacter oxidants, and P carotene is produced member of your Gynophoridaceae family of phycomycetes. And amino acids, some such like E. coli and cloning vector PBR322 were used to increase the genes for the production of amino acids. So like glutamate, L-lysine, mostly used at feed and food additives. So this is the golden rice technology. You can see Erwinia bacteria here being producing genes. So they are like green, red, and yellow. They are producing plasmids, and so we produce them into the plasmids, and then we transfer them into agrobacteria. You know this agrobacterium tumefaciens transformation of your plants. Then they are being produced in your embryo of kernel, and then hence you get golden rice at the end, that with having provitamin A producing rice embryos. Some of your students are also writing review about it. So enzymes are biological molecules that catalyze increase the rates of chemical reactions. Recombinant enzymes playing vital role in the food industry. So amylases from fungi and plants for making high fructose corn syrup, and proteases are used by biscuits manufacturers to lower the protein level of flour. So double-stranded plasmids, DNA expression vector. So promoter sequence is there. So you cut this DNA with friction nucleases and insert amylase coding DNA sequences into that and introduce into the recombinant DNA into your cells. So you have overexpressed messenger RNA or overexpressed amylase. So these both could be part of your game. Then there is a production of cheese with the help of rennet, chymosin, which is derived from the rennet used to manufacture the production of a cheese. Amino acids and peptides that are used by the yeast for fermentations, and proteases split the polysaccharides and proteins in the malt. So prochymosin genes are isolated from young calves and then they are transferred to plants, and this plasmid is then introduced into the microorganisms. So on expressions, prochymosin is activated into chymosin, producing 100% pure chymosin. And first step is the milk clotting process in cheese making, in which K-sinolytic enzymes contribute the vessel precipitation because of its specificity towards K-casein is best enzyme for this purpose. This is cheese production using recombinant chymosin. You isolate this cow from chymosin gene from the cow. Then plasmid DNA is cut into the restriction enzyme. Then chymosin DNA with plasmid DNA is being produced with and we join them together with ligase. then put into the bacteria. This recombinant DNA. Then chymosin protein is expressed in the bacterial cells, leading to the production of isolate chymosin protein. And hence we have isolated chymosin used to make cheese at the end. So DNA, exons, introns, having this exons 9, introns 8 are being produced like that. Then biofuel industry, cellulase used to break cellulose into sugars that can be also fermented. In the molecular biology, restriction enzymes, DNA ligase, polymerases used to manipulate DNA. In genetic engineering, important in the pharmacology and agricultural medicines, which are used in forensic science. On that note, thank you very much for being on this 25 or 24 days journey. Today is 23rd. On this MBBT course. So I'm with heavy heart says thank you to everyone for being part of it. Hope you have learned things that you came for. I'm also like bringing analytical technique part 2 in biotechnology that we use. So let's see. I'm trying to make new PPTs. So something about UV spectrophotometer, more about your centrifugation techniques, something latest, more other techniques I want to add in. So let's see how things works out. We'll let you know. So let me discuss with my admin and we'll let you know how we will conduct the exam. Let me discuss with them. So thank you each one of you who's been there, who is watching my videos later from the group. So you are our students who have given us your time in the evening 7 to 8 from the 30th January we are working on. I don't know, on 2nd of Feb maybe. And there was some 1 or 2 days that I was not able to take your class, but most of the time we were able to do so and everything went well. I hope you learned something, because remember, whenever you are going for any job or anything in your life, so these techniques will help you to crack any interview, to crack any job profile, any job description. Because if you know these techniques of biotechnology and molecular biology and biochemistry, they will really help you to reach your goals, to get your things that you're looking for. My best wishes with you, my all healthy blessings with you. God bless you, each one of you. I wish I could have like a Google meet with all of you. We can see each other and talk a little bit about the end, but I think time will not let us do so. Yeah. I don't know how many of you are spiritual or not, but on leaving you, I will send you this. Let me play one thing. It's from the. I was at the ISKCON temple recently, and there's a Prabhupada's kirtan going on. So maybe I play a little bit about that. It's not working. I don't know why. Anyhow, so thank you each of one of you who's been there, and yeah. If you have any questions, you can ask me on the anytime on that thing, Telegram, and yeah. Just keep learning, keep growing yourself, never stop. Read others' books also, and keep going on, keep going on, keep going on. No one can stop you. No one can stop you. No one can stop you. Okay, bye bye. Let it be 20, then we leave. I don't know what to say else. Yeah, Bipla, course is finished. Today is finished. We have this all presentations, all topics are being cleared. Everything is over by today. Yo, take care. And just want to say one thing before I leave. According to my experience of my 30, I'm 33 at the moment. And when I came to the spirituality, first I understand that there is a Vaheguru, yeah. Then slowly that Vaheguru let me show more about Hinduism, like Shiva, Krishna, Hare Krishna, Om Namah Shivaya, and then slowly I understand about Allah, that is universal God, and then at the end I came to conclusion that there is a super natural power, and that is present in everyone, in each one of you. It's just we need to recognize that, and that is God actually. So it is it's present in unconditional love, first of all, where there is a lot of joy, happiness, no jealousy, no hindrance, no bad thoughts, no lust, no anger, yeah, and yeah. So at the end, it came out to be one. It's one, and keep sending me your articles. Yeah, I will look for them, and yeah, that's from my side. Thank you very much.